Welcome to Peking University MOOC, Bioinformatics, Introduction and Methods. I'm Liping Wei from the Center for Bioinformatics at Peking University. Let's continue with our week 9 lectures on ontology and identification of molecular pathways. In Unit 1, let's look at ontology and the gene ontology. You may still remember this slide, which I showed you in week 1's lectures. The informatics in bioinformatics refers to the computer and computational technologies that we use to address biological questions. It follows the theme of from data to discovery. In the past few weeks, Dr. Gao and I spent most of the time telling you about many bioinformatic algorithms, software tools, and web servers. They are all based on biological data and operate on biological data. How can the data be organized so as to facilitate computation? This is the subject of this unit. If you think about it, biologists really don't make it easy for computers. Take the example of a human gene that many of you are familiar with, the Wnt1 gene. Some people, however, write it as Wnt-1. But a computer cannot automatically know that Wnt1 and Wnt-1 refer to the same gene. Even worse, Wnt1 was previously known as Int1. There's no way for a computer to know that Int1 and Wnt1 refer to the same gene. Wnt1 is a shorthand for winglets type MMPV integration site family member 1. The poor computer is really getting confused. Wnt1 is a member of the winglets type MMPV integration site family, but a computer looking at Wnt1 and winglets type MMPV integration site family has no way of knowing their relationship. To complicate things even further for the poor computer, the Drosophila research community have a habit of naming a gene after its mutant phenotype. So the homologue of Wnt1 is called winglets in Drosophila. You may be able to guess it easily, but it is very hard for a computer to guess things like this accurately. Another example is the highly conserved delta DNA polymerase. It used to be called CDC2 in yeast and now renamed to PO3. It is called DNA PO-delta in Drosophila and PO-D1 in mouse and human. How can a computer know that all these different names are referring to the same gene? A computer is only as smart as we make it to be. So if we want our computer to use its power to compute for us, we need to clearly define the data for it. We need to define all the entities, give each entity a unique name, enumerate all its synonyms and acronyms, and describe its properties. We also need to define the relationship between the entities, such as Wnt1 is a Wnt family member. Basically, we need to define a vocabulary. Not just any vocabulary, but a vocabulary that is controlled, dictating the use of predefined, authorized terms that have been pre-selected by the designer of the vocabulary in contrast to free natural language vocabularies. The vocabulary is common, meaning that it is agreed upon by different people to use it as a common language across different applications. Last but not least, the vocabulary is hierarchical, defining the hierarchical relationship between the entities. When this is done in a very formal way, this vocabulary is called an ontology. When I was doing my PhD at Stanford, I learned in the bioinformatics algorithm class that my advisor, Russ Altman, taught that an ontology was a specification of a conceptualization. I still remember very sophisticatedly every time I say these long words. To put it in more easy to understand language, an ontology can be defined more operationally as a set of concepts within the domain defined by a shared vocabulary to denote the types and properties of the concepts as well as the relationships between the concepts. Ontology had a long root in philosophy since the 19th century, before it was adopted in computer science in the mid-20th century. In philosophy, it is defined as the study of the nature of being, becoming, existence, or reality, as well as the basic categories of being and their relations. So why is ontology important? What does it enable us to do? First, communication. An ontology allows different people to communicate unambiguously. For example, with an ontology of gene function, different groups annotating different genomes can have a common language. Second, computation. An ontology represents knowledge in a computable form so that computer programs can analyze the data automatically. Third, discovery of patterns across different hierarchies. The hierarchical structure of an ontology enables us to go above a set of individual genes to find the larger functional categories or pathways involved. It gives us a bird's eye view. Open biomedical ontologies collect major ontologies in life sciences, including gene ontology, anatomical entity ontology, disease ontology, sequence ontology, systems biology ontology, and so on. The most widely used by far is the gene ontology. You may remember from our first week's lectures that since the first free-living organism was sequenced in 1995, more and more genomes were getting sequenced in the late 1990s. The yeast genome sequencing was completed in 1996, the C. elegans genome in 1998, the Drosophila genome sequencing in 2000, and the mouse genome was well on its way. In 1998, the genome scientists were busy annotating the genomes. They soon realized that a large fraction of genes were conserved across different species, but the homologous genes were often given completely different names in different species, making comparisons difficult. It made it hard to borrow knowledge from one species to aid the understanding of homologous genes in another species. To provide a unified common language for biology, in 1998, scientists of 3 model organism genome databases, fly-based, yeast genome database, and mouse genome database, decided to collaborate to create the gene ontology. The gene ontology defines a structured, common, controlled vocabulary to describe attributes of genes and gene products across organisms. Collaboration is key to build a consensus vocabulary. Today, the Gene Ontology Consortium has grown from the initial 3 genome groups to over 20 members, spanning multiple large genome sequencing projects. The gene ontology is divided into 3 categories to describe the genes and gene products from 3 different angles: molecular function, biological process, and cellular components. Each category is a hierarchical controlled vocabulary. The molecular function describes the elemental activities or tasks performed by individual gene products, for instance, carbohydrate binding or ATPase activity. The biological process describes the biological goals or objectives that are accomplished by ordered assemblies of molecular functions, for example, mitosis or purine metabolism. The cellular component describes the subcellular structures, locations, and macromolecular complexes that the gene product belongs to, for example, nucleus, telomere, and RNA polymerase II holoenzyme. Each of the 3 categories of the gene ontology can be represented as a directed acyclic graph. The 3 categories are the 3 roots, part of the biological process graph is shown here. Each node of the graph is a concept. For instance, the node pigmentation during development is a concept of a biological process. The edges between the nodes represent the relationships between the concepts. For example, pigmentation is a special kind of pigment. For example, pigmentation during development is a special kind of pigmentation. So you see, this is an edge pointing towards pigmentation. The graph is directed because the edges or relationships have directions, such as A is part of B, in which case B cannot be part of A. The graph is acyclic because all the edges point towards the root, so they cannot form loops. How can this graph be stored in the computer? Please pause for a moment to think about how you would want to do it. There are several formats for storing the gene ontology. One of the most widely used formats is the OBO format. Gene ontology currently uses OBO version 1.2. Here, each concept is named a term. Each concept has a few properties defined, including a unique ID, a name, a namespace, which specifies which of the three categories the term belongs to. Def is the definition of the term. Synonym lists all the synonyms and acronyms of the name. Finally, there is the definition of the concept's relationship with other concepts, such as is-a. Here is a real example of a part of an OBO data file. The first line is term. The next line describes the unique ID, which is CO000001 here. The next line specifies the name of the concept, mitochondrial inheritance. Next, the namespace, biological process. The definition of the concept is included here. And the synonyms and acronyms are listed here. Mitochondrial inheritance and mitochondrial inheritance are very similar to human eyes, but if you don't define it for the computer won't know what they refer to. The relationship between this and other concepts are specified here. Each concept can have relationship with multiple other concepts. Here, mitochondrial inheritance is a kind of organelle inheritance which has an ID of GO004838, and it is a kind of mitochondrial distribution which has an ID of GO00483111. There is an empty line between two terms. As you can see, such a standard format makes it very easy to write a computer program to parse the data. Another popular format for storing ontology is the XML format. The XML format is used not just in bioinformatics, but also in many other disciplines. It's a commonly used data storage structure. Each concept in the gene ontology is called a Go term. A term has an accession number which was like the ID in the OBO format. The term has a name, synonyms, and a definition just like in the OBO format. The relationship with other terms are specified here, such as is-a. Finally, the term is cross-referenced to related entries in other databases. Here is a real example of a part of a RDF XML file, which is a specific type of XML file used in gene ontology. Each term starts with <sign GO term. The URL of the corresponding entry in the online gene ontology server is listed here. Each term ends with </ GO term, the greater than sign. The slash signifies the end of it. You may notice that this structure looks very similar to an HTML file as well. The geo-accession number is given here between go-accession and /go-accession. The name, synonyms, and definition are given here. The relationship to other terms are specified here. In the second term shown here, cross-reference to its corresponding entry in the Interpro database is specified as IPR 009446. The definition of relationship deserves a closer look. The first relationship is a. B is A means B is a subtype of A. A is sometimes called a mother or parent node, and B is sometimes called a child node. For example, mitochondrial inheritance is an organelle inheritance. Pigmentation during development is a pigmentation. In the directed acyclic graph, the is-a relationship is shown with the eye on the edge, with the arrow pointing to the mother node. The second relationship is part of, such as B is a part of A. For example, ribosomal large unit assembly is part of ribosome assembly. Pigment metabolic process during pigmentation is part of pigmentation. In the directed acyclic graph, the part of relationship is shown with the letter P on the edge, with the arrow pointing to the mother node. The third relationship is regulates, such as B regulates A. There are two sub-relationships, positively regulates and negatively regulates. For example, the R on the edge specifies that regulation of pigmentation during development regulates pigmentation during development. The R is on a black background. An R with a green background on an edge specifies that positive regulation of pigmentation during development positively regulates pigmentation during development. Yes, you guessed it. An R with a red background on the edge specifies that negative regulation of pigmentation during development negatively regulates pigmentation during development. Once the relationships are defined, you can reason over them, and more relationships can be deducted. If A is a B and B is a C, then A is a C. If A is a B and B part of C, then A is part of C. If A is part of B and B is part of C, then A is part of C. If A is part of B and B is part of C, then A is part of C. If A is a B and B regulates C, then A regulates C. If A regulates B and B is a C, then A regulates C. If A regulates B and B is part of C, then A regulates C. Similarly, if A is a B and B positively regulates C, then A positively regulates C. If A positively regulates B and B is a C, then A positively regulates C. If A positively regulates B and B is part of C, then A positively regulates C. If A is a B and B negatively regulates C, then A negatively regulates C, and so on. Gene ontology is a very useful resource. All the data is freely available online at this website. As of December 2013, gene ontology has about 40,000 terms, covering 2,800 species, with over 573,000 genes annotated. The gene ontology defines about 76,000 relationships, 83 of which are is-a relationships, 8% part of relations, and the remaining 9% regulations. The deepest branch has 12 levels. You can browse and search gene ontology from the Amigo tools on the same website. Browsing can be done in a tree-like structure. You can expand a mother node to see all its child nodes. You can clearly see the relationship between the nodes. You can search for geoterms. You can also search with the gene name to find out about its geo annotation. Later in the lectures, we will talk about how to associate the gene with geo terms. Here are some summary questions for you to think about. See you at the next unit.